get that away. Okay. So I want to first start by talking about the outline by looking at some stuff that you turned in your TBE list. And I saw that most of you have turned in TBE lists, and I have not looked at them yet. The next couple of days, I will get a chance to look at those. So it asks you to put in your data figure and the legend if they're separate files, and that's so I can see those, and not toggle tabs and toggle windows every time. And then you've got a set of questions about what are the authors actually researching here. So an example of the big question, like for Connors, it is periodontitis and brain degeneration. Like if you had to say in a sense, what is this actually all about? And then I'm asking you a narrower version of that question. What's the targeted research question? And that's kind of like, what is your paper asking that's a little bit narrower than the big question? And so, like, example for Connors, is the link between periodontitis and brain degeneration actually happening in the gut? Something like that. So it includes the big question, but it's narrower. And then the specific question is coming from, what is your data figure actually asking? So for Connors, and I know his the best because he's doing this for seminar, and we've been talking about his a long time. His targeted question on his data figure is pretty much, are tight junctions messed up in the gut? And you probably wrote something better than that on that question, but that one is like based on the data figure. What is the experiment in the data figure actually asking? So those three questions in your outline are going to fall kind of into the introduction section. So when you write your short paper describing this data figure and how they accomplished it, you need a little bit of intro to like the major problem, the general research in this area, and then you're going to move into the specific question that the data figure is answering or is trying to answer. So that will be in your background section or your intro section. And then you are going to go into writing actually about the data figure itself. So normally this part is maybe a page, maybe one big paragraph, something not too long. And then you need to talk about data figure. So this question 6, listing the abbreviation in terms, this is not going in order of your outline. That's just kind of like a starting place. What's complicated about this figure? Make a list. So you won't really put a section lining up with this at this point in the outline. But you will need a section in your outline to talk about the method. And methods are kind of going to be second, usually, in how you describe these. So I'm going to come back to what's missing there, because it'll make more sense as we go through the rest of this. So you're going to talk about what the method is and generally how the method works. How it works. And then toward the end of talking about the data figure, after they have used the method and they have gotten the data, most of your figures have some type of statistical thing. I think yours does not. Like you've got a bar graph.
Yeah.
So you may not have to talk about the statistics, but most people have some type of statistics. Brooklyn, yours may not have statistics.
I couldn't tell you.
I'm trying to think about like what your figures are, but. So the statistics are going to come last. And because you can't do statistics until you have data. So they have a method, they're probably doing statistics. So what's left and kind of what is the most, needs the most decision-making and thinking from you guys is how do you take the abbreviations and the particulars of the experiment and describe that to somebody? So that section is kind of maybe 50% of what this paper turns out being. How do you describe what they did for the experiment? So that's going to include the experimental design, and it kind of goes with the methods. So this is where you have, this is where you have to think about what order would you write about this stuff. So they're using methods, they have an experimental design, maybe they had 5 mice per treatment, you know, something like that. And the other key part of this particulars are going to include the abbreviations. I don't feel super articulate this morning, but what's an example of an abbreviation that's really important for your figure? Somebody remember one?
Fish.
Fish? Yes, okay. think. Fish. Okay, so fish is fluorescent in situ hybridization. And you would kind of want to explain what that is and generally how it works. What's the point of them doing fish?
So I have to like see the gene or something like that.
They want to see the order of how those genes are compared to the centromere. It took me like 20 minutes to figure that out, but that's what the point of it is. And so something else that you would need to describe in that one is what are those genes and why do they care about those? So this is the creative part and thinking about, okay, assuming someone knows nothing about this, how would I explain it in a way that makes sense to them without writing a 20-page paper, but giving enough context to like understand what the figure is. Like Cassie, for yours, you will need to describe in some way, like they had people separated into high fiber diet or something like that. Mm-hmm. And then at the end, they're doing a comparison of how this related to different, whatever was in those bar graphs. So does that kind of help with your outline? So it's not writing a paper, but it's kind of putting in order, taking your abbreviations and methods, and the statistics are going to come last. I can definitely tell you that. But kind of thinking about telling a story in a certain order that will make sense to people. And then the other part of this assignment is to put in, oh yeah, tell me what, well, this is the TBE list. You already turned that in. I want to talk about the references. So in your assignment for turning in the outline and the references, let me go to that actual assignment. There we go. you're gonna upload a Word document, and I'm looking at the order. So I'm gonna be comparing this with your TBE list and looking at the order. And if I have a suggestion about changing the order, I will let you know. And then give me an idea of what your references are. I'm not gonna be grading your reference format right now, but I would like you to try to get them close to a scientific format so that I can see what they are. Don't put web links where I have to follow 10 web links and see what that is. So put the whole reference. I'm not going to be ticky-tack right now about the commas or the italics or how you have the journal edition listed, but just let me see what they are. And then this is so I can make suggestions if you need changes. Any other things you're wondering about that? Okay. And these can be due next Tuesday. It says Thursday, but I'm a little behind grading TBE list, so these can be due next Tuesday, which would be October 29. Yes, October 29. And go ahead and change that in here. So if I don't do it now, I will forget. Okay, so we will wait and go over exams on Thursday because I don't want to have that on video and I don't want to process 2 videos. So we'll do that on Thursday. So we're going to start into a new unit now. And this one is containing a lot of the key material that should be in a cell biology class. So unit 1 is kind of review-ish stuff. And unit 2 is also kind of review-ish stuff like glycolysis and electron transport chain, kind of delving into further details. But this section tends to be stuff that is actually, you've never heard of it, and actually it's the heart of cell biology. So this section is going to continue kind of talking about the life story of a protein. The life story of some type of protein. And every protein, we have a gene in the DNA to build it. It has to get transcribed. It has to get translated. It has to fold up into a certain shape. And what we're talking about next is how does the protein get to its correct location? How does it get to its correct location where it is supposed to be doing a job in the cell? So that question is for today and Thursday. And then the following week, we move into cell signaling and then cytoskeleton. So those are the topics in this particular unit. And we're going to start with this question: how does the protein get to its correct location? And I like to start with this picture from the old book, which is showing all the parts of the cell and kind of how they were situated, and describing now not the organelles and the structures that pop out at us, but something in the background, which is the cytoplasm. So on this picture, cytoplasm is the light gray. And our 7th grader just started a unit about cells. And his brother was actually helping him with it. It's like, this is amazing. This is amazing. But one thing that I know that kids learn the name cytoplasm and then they never talk about it again in the intro because it's like, move on and talk about a mitochondrion. It's a powerhouse of the cell. The cytoplasm and how it acts actually is really key to understanding cell biology. And so we can think of cytoplasm as everything in the gray, but there's also something called cytosol. And actually, cytosol is what's in the gray. So cytoplasm includes the cytosol, which is in gray, and everything that it contains. So the organelles, ribosomes, anything big, and also cytoskeleton that is assembled. And I've mentioned this in some way before, but it's important to think about cytosol is an aqueous solution and it acts like it. It's liquid and it has soluble components that float around and diffuse around in it. And if something is not tied down or bound to something else, it can diffuse through the cytosol anywhere in the cell. And that's really key to understanding how cells work. So every protein in the cell has a particular destination, which could be going into an organelle, it could be going out to the plasma membrane, or it could be floating around soluble, diffusing in the cytosol. And getting to that correct location is really important for the health of the cell overall. So those are some of the possible locations. Where could the protein be going? So we're going to be talking about not every single one of these places in the cell, but about half of them. So definitely nucleus. How does a protein get into the nucleus if that's where it's supposed to work? How does a protein get into the endoplasmic reticulum if that's where it's supposed to work? How does it get into a membrane? Or how does it get totally out of the cell to be secreted? So we start with nucleus and endoplasmic reticulum today as destinations for proteins to do their work. Okay, so talk to your friend or think to yourself about one protein or one molecule that needs to go into the nucleus and one thing that needs to come out of the nucleus. And we'll make a list. What needs to go in, what needs to come out of the nucleus. What's something that would need to go into the nucleus? I'm going to put the cartoon back up. What's going on in there that needs workers? Replication. The reason I'm smiling is that when DNA replicates, the nucleus has kind of fallen apart. But I like that answer. That's something that happens with DNA before the nucleus falls apart during mitosis. What else would happen inside that nucleus without the DNA and the nucleus having the big disruptive mitosis? Transcription, yeah. So one thing that needs to go into the nucleus are transcription factors. RNA polymerase pieces would need to go into the nucleus. Something like polyadenylase would need to get in there. So those are a few examples. We need proteins that accomplish transcription to move into the nucleus to do that work. What's something that comes out of the nucleus into the cytoplasm? There's not as much, but some important things. mRNAs, yeah. And that's the major one that we're going to remember for this class is that, okay, you've got transcription going on inside the nucleus and you need workers for that to go in there and do that work. The messenger RNA that they make is supposed to come out into the cytosol so that ribosomes out here can translate it. So lots of things need to go in, some things need to come back out, and how that happens is what we're going to look at next, and what is surrounding the nucleus to allow that transport. Okay, so I think I want to come back to this picture. I think I've got it listed twice in the slides, and the later place is going better with what I want to. Well, no, we got to talk about it here. Okay, so what we're looking at here is we've got messenger RNAs that are in the cytosol, and they have a 5' cap, and they have the 3' poly A, which is not shown, and they hopefully have been spliced appropriately. So splicing factors could also be something that needs to go into the nucleus. And every protein starts being translated in the cytoplasm on ribosomes that are free or ribosomes that are soluble. So if you quickly got these slides this morning, you could add the word soluble right there. They all start translation on a soluble cytoplasmic ribosome. And where the proteins eventually need to end up is different based on their job. Some of them need to go to the nucleus. Some of them need to go into mitochondria and chloroplast. Some of them need to go into the inside of a peroxisome. You've got this picture. Peroxisome is listed twice. This is the interior, talking about the interior of a peroxisome. But there's other proteins that need to go into some type of membrane. So this set I would kind of summarize as these need to go into a membrane, or they need to go completely outside the cell, or eventually into a lysosome. So there's kind of two major routings of proteins into nucleus or interior of those organelles, or go to a membrane somewhere in the cell, totally leave the cell, that goes with secretory vesicle, or go into a lysosome. And if a protein is heading to these destinations, they will have to first go into the endoplasmic reticulum. So if proteins are going these places, or these places, they must pass through the ER as a part of their life journey, let's say. And that concept is repeated somewhere in these slides. But the question of how does a protein know, or how does a cell know what's the destination of a protein? Where's it supposed to be going, and how do I start getting it there? That is accomplished by proteins having signal sequence. So keyword on this slide is signal sequence. And a signal sequence is a piece of a section of the protein that's got certain amino acids that serve as a destination tag. And the point of this slide is not for you to memorize what the signal sequences are, but to see what type of places they can make a protein go. So there are several key things I want to use this figure to show, and the new book did not have something quite like this. So signal sequences can be at the C terminus. Change this to N, let's put N first. N terminus, or they can be internal to the protein sequence, or they can be at the C terminus. It depends on what type of signal we're talking about. So signals to tell a protein to go into endoplasmic reticulum are N-terminal. That's what this means. So at the N-terminus of the protein, the signal is early. Some signals are C-terminal, so they come right at the end of the protein chain, and they have a certain sequence. This one's N-terminal. This one is internal, not N-terminal, but internal in the middle of the protein, and so is this one. So if a protein is supposed to be going to a certain destination to do its work in some of these places, it's going to have pieces of the protein that give shape information and give four-force interactions that serve as tags to go to these locations. That's how they know, that's how they get targeted to the correct location, is through signal sequences. And we're going to start with nuclear import. How does a protein get into the nucleus and get to that correct place? These are some examples of nuclear localization signals. So that's a signal sequence that is targeting a protein specifically to nucleus. And they are internal in the middle of the protein primary sequence. So here's an example of one, and it's displaying these amino acids on the surface of the protein. And this one is all in one stretch. All the signal sequence come together. This one, they're kind of separated by distance, but they're close together on the surface. And they tend to be the basic amino acids. And when we see in a few minutes K and R, if you specify these amino acids by the one-letter code, they become a lot of Ks and Rs together as basic amino acids. So these proteins that are going to the nucleus, they get translated completely in the cytoplasm. Whoops, yes. They get translated in the cytoplasm, and they finish translating, and they fold up, and they are going to have a segment on their surface that is going to bind to another protein that helps them go into the nucleus. So they finish folding, they've got a basic patch on their surface that's going to bind a helper protein to help them get into the nucleus. So the way that they get into the nucleus is they have to get past a lot of barriers, let's say. So now we're going to look at what's the structure of the nucleus actually. So this is the structure of a nucleus during interphase. During interphase. People in genetics, are you talking about mitosis stuff these days or not yet?
We already did.
You already did, okay. Most of cell biology, until we get to right before Thanksgiving, I'm always talking about a cell in interphase. When you study mitosis, interphase is the boring part, but interphase is, for the most of a cell's life, the useful part. So during interphase, when the nucleus is intact, it's got double membranes around it, and those membranes are actually continuous with the endoplasmic reticulum membrane. Is that new or is that old? Good. I'm glad it's old for you. Somehow I was not aware of that until I started teaching this class on it. Really? Because I fell asleep in my cell bio class a lot in college. So double membrane, actually continuous with the ER. And in the section that is surrounding the DNA, which would be an interphase relaxed and decondensed, there are some pores. And so we're going to be focusing next on what's the structure of these pores that let things in and out of the nucleus. And they have so many parts, they're called nuclear pore complexes. The other part of nuclear membrane and nuclear structures that are going to be important 3 different times in the rest of the semester are called nuclear lamins. Nuclear lamins. Nuclear lamins, I think of as like tent poles. So they're holding up the structure of the membranes around the nucleus and providing structural support. So I'm going to toggle back a slide and show where would these be. In this picture, it's this stuff here. This network of fibers that's kind of supporting the membranes all around the exterior periphery of the nucleus. Nuclear lamins are actually part of cytoskeleton. And we talk about cytoskeleton at the end of this unit, but the lamins individually have this barbell type structure, and they form non-covalent quaternary structure with each other. So single one joins with a different one, they twist around each other to form a dimer. And then 2 dimers associate in this manner to form a tetramer, and they form a filament. But all of those associations are non-covalent and via the 4-force interactions. So they can assemble or they can disassemble. And during interphase, when the nucleus is intact, they are assembled into a grouped network called nuclear lamina, the tent poles. Okay, so now we have a big zoom in. Oh, I walked back here. You can reach that. Thank you. Tall people can reach far. Okay, so now we zoom in, and here in the yellow in the background, those are lamina. That is the lamina with individual nuclear lamins. And here we've got the outer and inner membrane of the nuclear membrane. And this is the pore, nuclear pore complex. We have also got relaxed chromatin DNA right up against this. So there's not like a space and then the DNA starts over there. It's pretty crammed in there, but relaxed when the nucleus is intact. So here is the nuclear pore complex, and it has a lot of parts to it. And I need to describe some of them on this picture because this picture is big and it's got space to do so. Well, I don't know. I'll go forward. I want to see how the other picture looks. Okay. So these nuclear pore complexes are spaced kind of at fairly regular intervals around the periphery of the nucleus. They are not actually this close together. These are artificially purified and assembled ones. But it's showing, if you were to look down this hole at a single nuclear pore complex, it's got an 8-fold symmetry and they're kind of pretty. They look like BB gun caps, actually. And then if you look at them side on, they're actually dense enough that they'll show up on an electron micrograph, and you can see that they're kind of pinching the two membranes together at this passageway. So this picture is showing the different parts. And so I need to remind myself what are the correct names of the parts before I go back to the big picture and show them on there where they're easier to see. So let's go to this picture, and Connor, if you would turn the lights on so we can see what we're gonna write. Okay, so make a note right here that these wavy extensions going on the cytosolic side are called cytoplasmic fibrils. And they kind of wave a little bit. They have a little bit of flexibility to them. And there's 8 around the ring, cytoplasmic fibrils. Down here, this apparatus would be actually in a circle. So all of this is in a circle or a tunnel in 3D shape. And this is called nuclear basket. Nuclear basket. And then if you have this picture, the big one, if you would draw in some wavy lines in here through the pore and call those IDR, which are intrinsically disordered regions. IDR. And the IDRs kind of act like filters. So you've got some long, slightly bendy cytoplasmic fibrils facing the cytoplasm. You've got a network of flexible, intrinsically disordered region in the middle that's acting like a filter. And then you have nuclear basket on the nuclear facing side. Together, those prevent diffusion of most big molecules in and out of the nucleus. So nuclear pore complex in total prevents diffusion of most larger molecules in either direction. So if you are a protein that needs to get into the nucleus to do your job, you cannot rely on diffusion to get in there. You have to be escorted or chaperoned or guided in there by proteins that have qualifications to pass through this complex, this nuclear pore complex. Water can go in and out, oxygen can go in and out, CO2 can, but it is surprising how many things can't because there is just so much in the way. And to me, this has always looked like a Frisbee golf basket. Like if you put a few little things on the top of the Frisbee golf basket, but you're not trying to throw things in from the side, they need to go this way. So that's where we're gonna have our break. And that also helps in a long class for me to break up the video into a loadable size. Oh Lord, I don't want to read my transcript. Hang on a second, I'll tell you a terrible story.